So Alfred Hershey and Martha Chase show that DNA is the genetic material of T2 phage. And here I have to tell you a little bit more about virus and bacteria, and especially type of virus that attack bacteria that are the bacteriophages. And that was the key model organism that allowed us to study this. Another key component was the use of radioactive elements that allow them to mark and label the bacteriophage. So let's talk a little bit about that. So as I said before, we just talked about this very interesting experiment, and although they found that the transforming substance was DNA, many biologists remain skeptical because little was known about DNA. So it has been a long time since the DNA was discovered, and there have been different lines of evidence showing that the DNA was the genetic material. It was very difficult for most of the scientists, most of the biologists at that time, to consider that it was DNA, the genetic material, not proteins, as I said before, and as we explained in the previous slides. So now I'm going to show you a very interesting model organism that is the phage2, and they are going to show you the reproductive cycle that is fundamental to try to understand the experiments of Hershey and Chase.
Bacteriophages, or phages, are viruses that infect bacteria. This is a T2 phage, which consists of DNA inside a protein coat. The reproductive cycle of the T2 phage begins when the tail fibers of the phage stick to the receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes are used to manufacture phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures, or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the reproductive cycle of bacteriophage T2.
So now that we watched the video, we know that more evidence for DNA as genetic material came from a study of these bacterial viruses. Now we know that they have a head, a tail, and that they have these tail fibers, and that they also have DNA inside of here, and they can infect bacteria, and that viruses and or bacteriophages are used for genetic research because they have this amazing property that we can insert any DNA into a bacteria. And we also know that a virus is DNA or sometimes RNA enclosed in a protected coat, usually made of proteins. Now I'm going to show you this experiment that I think make things much more clear than me just talking about how this experiment takes place.
In the Hershey-Chase experiment, bacterial viruses, called phage, were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing p32 labeled deoxyribonucleotides. This resulted in a phage population with p32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the p32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
So as we watch in the video, and I think that is very clear, I'm just going to repeat small pieces to make sure that you understood. This experiment allows us to understand that not proteins, but DNA was in charge of transfer the genetic material. And I remind you that, well, the DNA has phosphorus, yeah, it has a phosphate group, but it doesn't have a sulfur group. That in that way, the DNA, that label, that radioactive label, can differentiate between DNA and proteins. On the other hand, the proteins can have sulfur, yeah, and in this case, and it doesn't have, in this case, it doesn't have a phosphate group. So it was a perfect way to differentiate one was DNA and which one was protein. So as we watch in the video, when the bacteriophage invade or infect the bacteria, and later you use the blender to remove the fakes that are not inside of the bacteria, later what is going to happen is that the virus is going to attach to the DNA of the bacteria in both cases, but in one case you're going to have that the progeny of the fakes that contain phosphorus are going to appear, while the ones that don't have phosphor, the radioactive label. And in that way, we can show that mainly DNA, but not proteins, is inherited from the parental phage. Now, to summarize all the information that we have seen, I'm going to show you a video that it will talk about these key points that I just explained.
The Cold Spring Harbor Laboratory was the site of our next great discovery as well. It happened here in 1952. Biologists Alfred Hershey and Martha Chase were studying bacteriophage, a virus known to infect bacteria. The virus takes over the bacterium's internal machinery and commandeers it to produce more viruses. Bacteriophage consists of two simple components, a protein shell, and inside, a mysterious substance scientists had known about for decades called deoxyribonucleic acid, or DNA. Hershey and Chase wanted to know whether it was the protein or the DNA in the virus that carried the genetic information. Using two radioactive chemicals, they labeled the protein in one set of bacteriophages and the DNA in another. Then they exposed a culture of E. coli bacteria to these viruses. After using a kitchen blender to separate the bacteria from the empty viral protein shells, Hershey and Chase saw that the radioactive protein was not penetrating the bacterial wall. But inside the bacterium was the radioactive DNA. It was the DNA, not the protein, that was able to direct the host cell to make new viruses. It was an extraordinary breakthrough. Thanks to Hershey and Chase and the other scientists who helped pave their way, suddenly we understood the true identity of DNA. It's genetic material, the blueprint of life. And it all came out of a blender like this. Hershey was awarded the Nobel Prize in 1969 for his work, but Hershey and Chase's blender experiments went on to inspire a new era of genetic research.
Now I am going to I want that you see a small documentary about Watson and Crick and how they found the DNA structure, and that you answer a few questions.